And that seems like the perfect note to start off a set of notes with. A yawn. Hello, and here is the much promised cell cycle and mitosis video. So let's start taking a look at what's going on. So, um, cell division is if you are a single-celled organism, basically your entire form of reproduction. It is an asexual form of reproduction because there's no exchange of genetic material or no recombining genetic material. Instead, it produces clones. This is the method by which all single-celled organisms reproduce. In bacteria, in particular in prokaryotes, so bacteria and archaea, it's called binary fission. Binary fission is a much simpler process than mitosis, which we tend to go over um, which we'll go over later. Basically, you don't have a nucleus. You have your DNA centered in this region called the nucleoid. Because you don't have a separate nucleus, you don't have to worry about breaking it down. You also only have one single chromosome. And since you only have one single chromosome, you don't have to worry about did I get all the chromosomes in each daughter cell? There's just the one. So basically, the DNA attaches to a point on the cell membrane for each copy of their single chromosome. And then the cytoplasm starts growing in between. So you see this space has grown. And then once it gets to a certain point, it starts pinching inward. And that pinching inward is called invagination. And then eventually the two cells separate, forming a cell wall between them, and now you have two daughter cells. The process is much more complicated in eukaryotes. It is, you're going to have to do mitosis because of the multiple chromosomes, mostly. So let's look at the differences in chromosomes in prokaryotic versus eukaryotic cells. So in prokaryotes, you have a single circular chromosome. It may be kind of twisted up on itself, but it is still a single piece of DNA. It is replicated and distributed during binary fission. That's, whole process. It's a fairly simple structure, though even though it's a fairly simple structure, it can still have thousands of genes on it. On the other hand, eukaryotic chromosomes are linear. They are a single long line, and you even your simplest eukaryotic cells will probably have multiple chromosomes, which means you need to make sure each daughter cell or each cell that is produced gets every single chromosome that it needs to, which makes this process more complicated. In eukaryotic cells, you replicate the DNA, the chromosomes, during S phase, so well before you start mitosis, and then you distribute them during mitosis. Um, mitosis, it's nuclear division, redistribution of your chromosomes so that both daughter cells have every single chromosome. So, let's talk chromosomes. Most of the time, your DNA is going to exist in this kind of stringy, diffuse material called chromatin. Chromatin is somewhere in kind of this region. That's chromatin. Chromatin is more than just DNA. It's DNA with a bunch of proteins associated with it. So here you see some proteins that form kind of a core that your cell will wrap your DNA around and then kind of seal it with this orange guy, which is a histone. So you'll hear talk about histones, especially when we get into genetics. But for the most part, you don't tend to leave your DNA just kind of unwound sitting everywhere. You wind up parts of it that you're not using around histones, and then the parts that you do need regular access to tend to be kind of more spread out. But when you get to the point where you are trying to move these things around and move them from one part of the cell to another, it is a lot easier to do that if you condense them. So condensed chromatin is basically a chain of DNA wrapped around some nucleosomes and histones. Then that chain is coiled up, and then that chain is coiled up, and you just keep coiling and coiling and coiling until you get this larger structure that's actually visible under a light microscope called a chromosome. So it's all DNA, but depending on how it's arranged, what it looks like, we call it different things. Part of the reason why we don't just call them chromosomes inside the nucleus is because it'd be like this. I just drew 3 chromosomes. Can you distinguish between them? No. But we do use the term chromosome to refer to the set of genes or the set of DNA that travels together. But since we cannot distinguish individual chromosomes under a microscope like this, we just, some scientists were like, let's just call it chromatin. Because it's all that spaghetti looking stuff in the center. So let's look at some of the important structures on a chromosome. By the way, this X shape that you're seeing, that is a pair of identical molecules joined together at the center right here. That center part is called the centromere, and we're going to talk more about it on the next slide. The centromere is important for 2 reasons. One, it's where you join sister chromatids. So those are joined at the centromere. And then it's also going to be important because that's what your cells use to drag around your chromosome and pull it from one end of a cell to another. So you can call a chromosome either this single unduplicated chromosome, or a chromosome can be this pair of sister chromatids. Either one of those is a chromosome because here we're referring to the group of genes and DNA that travel together as a chromosome rather than like the physical structure. So if you see the single line with centromere like a bulge in the middle, that is a chromosome. It is just one copy of the chromosome. If you see like the X shape, that is 2 identical chromosomes joined together. You have 2 of them because you're about to separate them and give them to 2 different daughter cells. And you don't need to know anything about the arms. That's about it. Let's talk centromeres now. So centromeres are located at various locations. It depends on the specific chromosome. Some chromosomes will have the centromere very close to a tip. Some will have the centromere like smack dab in the middle of the chromosome. That's one of the things you look at if you're like identifying chromosomes on a picture. But these are not only where you have sister chromatids attached, it's also where the kinetochore is found. And the kinetochore is an important part of it. It is this region of proteins that attach to microtubules. And if you remember from when we talked about cell parts, microtubules are these basically long flexible tubes that you can build and break very easily because they're made of a bunch of repetitive subunits that you can just kind of put together like Legos and then take apart like Legos. So they're very good for moving stuff around the cell, especially. And that's what their job is here in mitosis. So you see on this detailed diagram right here, this is the microtubule. Basically what happens is the microtubule gets fed up through the kinetochore, which then breaks it down. So pieces go kind of scattering off into the cell. And as it does that, these little arms will attach further and then they'll basically contract, bringing that in. When they do that, they move. So basically, it's like a little monster almost, like eating and then moving up the microtubule and eating it away. So the kinetochore is important because that's where you have attachment. So if you have issues with mitosis with cell division, one possibility is it could be a problem with your kinetochores. So let's talk cell cycle. What's the purpose of it? Well, reproduction, but not necessarily reproduction of a whole organism. It is how we grow, at least for the most part. I mean, when you're getting from a small child to an adult, yes, you're growing by adding cells. And there are some cells in your body that are constantly reproducing. Your skin cells, for example, never stop reproducing. And then there's also regeneration. So if you are a lucky lizard and you lose a limb like a tail, you can actually grow that back. Arms and legs, not so much. So this is an overview of the cell cycle. It's divided into interphase, and mitosis. Interphase is divided, oh, here, I have that right there. Interphase is divided into several phases that you'll be responsible for knowing. Mitosis is divided into several phases that you're actually not going to be responsible for having memorized. You do need to recognize the names of the stages of mitosis, and I don't have it written anywhere else. So it's prophase, metaphase, anaphase, and telophase. You do not need to have memorized exactly what happens in those phases. You don't need to memorize diagrams. You need to memorize what's happening in general during mitosis, and you need to be able to recognize if a question asks you or starts giving you information about something in metaphase or something in telophase, that you're able to then kind of figure out what they're talking about. So, the multiple stages of interphase are basically two stages where you have growth and preparation for division. These are called G, which stood originally for gap phases, probably because when they were looking at this stuff under a microscope, they couldn't really see much happening. But we always or often think of it as growth. It's also preparing for cell division. So if you need more organelles, you need to make a bunch more endoplasmic reticulum. You need to make some more Golgi. Your mitochondria need to reproduce. You need to make some more ribosomes. All of that stuff is happening during the G phases. The S phase stands for synthesis. It's where you make a copy of the DNA. So between G1 and S phase, your DNA doubles. So if you were like graphing this, if this was G1, this would be the amount of DNA you have. If this was S, your amount would grow. Let's see. I'm trying to do this right. There we go. And if this was G2, you would have 2 times the amount of DNA that you have in G1. During S phase, the amount of DNA grows because you're synthesizing it, you're making it. So then after that, you've got gap 2, you're finally preparing for mitosis. That's when you make your final preps. Notice that there are these stars. These are cell cycle checkpoints, which we're going to talk about in a little more detail in a little bit. So there are some important structures during mitosis. Centrosomes are basically these centers of microtubules. They're also called, or you can refer to them as centrioles. And basically, microtubules grow from this. They create the other structure we're going to talk about, the protein spindle. In animal cells, there is a different component in plant cells called the MTOC, or the Microtubule Organizing Center. You just need to know that, see, Microtubule Organizing Center. You just need to know that that's what these structures are for. If you interfere with microtubule building, then you're going to interfere with production of the protein spindle. And the protein spindle is what's actually dragging around chromosomes. It's also got a role in helping to kind of push the kind of elongate the cell, basically. So these guys will grow and then push. So it'll push that way and that way. That helps push the cell apart, and then those microtubules will get shortened, because remember, this thing is basically eating away at the microtubule. As that happens, it moves further along towards the pole. So that's how it moves those chromosomes around. So the mitotic spindle, the protein spindle, is absolutely essential. Without it, you can't undergo mitosis because you can't move your chromosomes around. So mitosis is at its heart nuclear division. You're not going to divide up any of the cytoplasm during mitosis. That happens during the separate process of cytokinesis. Cytokinesis happens at the end of mitosis, so they overlap in time. But cytokinesis starts towards the end of mitosis. And in mitosis, you are just dividing up the chromosomes. And the major purpose behind it being such a complex process is that you want to make sure that each cell that is being produced is getting a copy of each chromosome. So there are some important moments in mitosis. Remember I said we're not focusing on the phases. We're focusing on a couple of important key events. The first one is when the kinetochores attach the centromeres to the spindle fibers. This is actually so important that after this, you have a checkpoint. Before you can move forward through mitosis, your cell will stop and make sure that it has properly attached microtubules to each of those kinetochores on either side of the chromosome so that they can be pulled apart. If that is not the case, if those have not attached correctly, the cell will pause and attempt to fix this. Then they're lined up to the center of the cell. That way, that's called alignment. That way you can very easily separate them. And then finally, the separation happens. So those kinetochores are going to eat up the microtubules, and the motor proteins that are in them are going to start pulling the chromosome towards one end of the cell. So you see here, this is a microscope image of a cell that is in the middle of anaphase, which is the phase where you separate. This is one grouping of chromosomes that will end up in one daughter cell, one grouping of chromosomes that will end up in another daughter cell. And one thing I hope you notice is basically this is one of those chromatids. Notice it looks like it's being pulled basically by that kind of end right there. It's being pulled by that center. And so the like rest of the chromosomes just kind of flapping around behind it. Because remember, your cell's chock full of stuff. Your kinetochores and everything, they're pulling this stuff pretty hard to get it to move through the rest of the cell contents. Also, keep in mind, for all of this to happen, notice there is no nucleus. In fact, these bits right here are probably fragments of the nuclear membrane. They're fragments of it because you want to rebuild it later, but you don't want it getting in the way. You basically use the area of the whole cell to try and do this. So there's still organelles around. We're just not drawing them in because we're focusing on what's happening in the nucleus. So cytokinesis starts at the end of mitosis. So notice we have multiple phases of mitosis here, and this is telophase, the last phase of mitosis. Basically, once you start separating those chromosomes, once they get far enough apart from each other, you can start the process of cytokinesis. It looks differently in animal and plant cells, and the reason for that is plant cells, if you remember, have a cell wall. Anything with a cell wall might have to undergo slightly different cytokinesis. In an animal cell, you can basically, I liken this to taking a very, very, very chunky person and tying a belt around the middle and then just pulling that belt and pulling that belt and pulling that belt. That doesn't do anything except for make the overweight person you are pulling a belt on very angry. But if you do that on a cell, that's actually going to be able to contract quite a bit because the cell membrane is flexible, and it's going to be able to pull that cell into two new cells. We call this place with that kind of belt of tightening proteins the cleavage furrow. It's basically a ring of microtubules that go around the cell, and it starts contracting. As it contracts, it squeezes the cell into halves, and then they're divided. You can't do that with a plant cell, though, because a cell wall would get in the way of that. So instead, plants basically just build a new cell wall and cell membrane between them. They'll make vesicles, which remember, tiny little bubbles, with all the components that make up the cell wall and the cell membrane, and they'll send those to the center of the starting to divide cell. At the end of anaphase, beginning of telophase. Those things will fuse and they'll form this barrier between the two cells. And then each of those cells can lay down materials here to make a new cell wall. Now, here's where we really want to focus. Why? Because, well, this is probably where you'll see more questions asked. Control of the cell cycle. We talked about checkpoints earlier. Here's your information about it. Because you want to make sure, it's very complicated to make sure that you coordinate all of your cells, make sure that the cells that you want to have divide are dividing, and the ones you don't want to divide aren't dividing. If all of your cells divided uncontrollably all of the time, well, one, you wouldn't have like you would just get bigger and bigger and bigger, and you wouldn't have any organization. Because for your body to work, you need a certain number of muscle cells. You don't need to keep growing that number. You need a certain number of brain cells. You don't need to keep growing that number. In fact, as you age, the number of brain cells you have decreases because your brain's like, yeah, that one's not working. Let's get rid of it. Sucking up energy. So this precise control of the cell cycle is mediated by checkpoints. A checkpoint is basically a point in the cell cycle where you stop and you check your work. That's kind of the big thing I liken it to. You stop and you check your work. At this point, for example, it's, did I have accurately replicated DNA? If I didn't, then I stop and I fix things until I move on. And then I can keep going with the rest of the cell cycle. If this is a non-fixable problem, then the cell can actually do something that cells sometimes do and undergo what's called apoptosis, which is programmed cell death. I know you guys remember from freshman bio that when you have uncontrolled cell division, that results in tumors and cancer. So in order to not have that happen, your body can take a cell that might become cancerous and trigger it to undergo apoptosis, where it dies and doesn't form a tumor. So let's look at these checkpoints. There are three. The G1 checkpoint is this one down here. It's basically asking a few questions. Is our DNA good? No damage? We're good. Are there enough resources? If there's not enough resources, there's no point in growing a new cell because, if you grow a brand new cell and there's not enough resources, something's going to die. Another thing that your cells look for, are there any growth factors? Growth factors are chemicals that signal other cells to grow. So cells can release growth factors to cells around them to say, hey, we don't have enough of us, make some more. And at this point, if you don't have these conditions met, or you don't have any growth factors, the cell can choose to stay in G0. G0 is just cell cycle arrest. You're pausing on the cell cycle, and your cell's just doing its thing. Just keep in mind, while DNA is being synthesized and copied, you're probably not using it to make proteins. While you have it all coiled up during M phase, you're not able to access your individual genes. It's like if you packed up a bunch of books in boxes in a van, and then you said, oh, wait, I wanted to check something on one of those books. Well, whoever's moving your books will be like, not until you get where you're going, man. Because everything is packed away. All right. Your G2 checkpoint is right before you go into mitosis. It is this guy up here, and it is influenced by a couple of things. Is our cell big enough? If it's not, we're going to stop and we're going to make some more cytoplasm or organelles. Is our DNA replicated correctly? If it's not, we're going to stop and we're going to fix it, because changes in DNA can be very harmful. And if not, then we pause. If all of those conditions are met, we move on to mitosis. Now, there is, like I said, a checkpoint in the middle of mitosis. It's after basically prophase. So what you're checking here is, are my chromosomes attached to the spindle properly? If they're not, and if I'm not able to divide up my chromosomes evenly, you pause. And then the cell works to get those sister chromatids attached to spindle fibers from opposite ends of the cell. All of this is controlled by these classes of molecules called Cyclin-Dependent Kinases. So cyclin-dependent kinases, or CDKs, are kinases. That ACE means it's an enzyme. The fact that it's a kinase means it phosphorylates things. It adds a phosphate group. And that phosphate group, depending on the target protein, could turn the protein on, could turn the protein off. It just depends on the function of the protein and the particular protein. So CDKs, when they're active, phosphorylate other proteins. If you remember from cell signaling, a lot of stuff happens because of phosphorylation. Now, in their normal form, just by themselves, CDKs are not active. In order to become active, they need a cyclin. A cyclin is another molecule. It's called that because the concentration of it cycles during the cell cycle. So it'll go up and down and up and down. Only when you have a cyclin present will a CDK be active. And each cyclin or each CDK has a specific cyclin it associates with. You have multiple cyclins and multiple CDKs in your cells. Some of them will be like making sure you get through your M checkpoint fine. Others might be making sure that you get through your G1 checkpoint fine. And so that way you can control different parts of the cell cycle. So cyclins are called that because they are their concentration is cyclical. At certain parts of the cell cycle, the cell will be directed, if everything's going okay, to take DNA, read it, make an mRNA, and make this protein, the cyclin. Then the cyclin and the CDK can bind together and become active. Usually, that will cause the phosphorylation of some other proteins that are going to help move the cell along the next stage of the cycle. Once you are through that checkpoint, the cell takes the cyclin and breaks it down by a molecule called a protease. Once it's broken down, the CDK becomes inactive again and is no longer able to tell the cell, hey, move on to the next stage of the cycle. But that's fine because it's done its job. If you are, waiting for the G2 checkpoint, that's a different cyclin and CDK that are mediating that. So this guy's job is just to get you through one checkpoint. You only want it to exist and be active if everything is ready to move on. If you have problems, you want your cyclin to not be made, or you want to degrade it, that way CDK won't become active, won't phosphorylate proteins, and won't move us on through the cell cycle. So here is a kind of animation. So we've got a nucleus, cytoplasm, that's all we're looking at. We've got an always present CDK. It is always there. It's not broken down. You just constantly have these hanging around in your cells. And this guy has an inactive form, or it's inactive because there's no cyclin. Then you have over here, you have inactive proteins. These are the proteins that would help move you through the cell cycle, but they're inactive. So at this point, this cell's not going through the cell cycle yet. But you can take that nice CDK, you can have your DNA being read to transcribe and translate something, which will synthesize proteins. In this case, the cyclin. That cyclin, which is inactive on its own, binds to the CDK and turns the CDK on. That CDK is then active and can phosphorylate other proteins, like those proteins that would help move through the cell cycle. Once that's all done, the cell is going to degrade your cyclin. Without active cyclin, without like actual cyclin, your CDK is going to be inactive again. See, inactive. And then those proteins that move you through the cell cycle will become inactive as well, because there are proteins that come around and will dephosphorylate these molecules. So that way you don't have a constant on signal. You have it turned on when you need it and then it very quickly turned off. One example is mitosis promoting factor, or MPF. MPF is the name of a combo of a CDK and a cyclin. When you have a high enough concentration of MPF, basically when you've got enough cyclin produced to make that active CDK cyclin complex called MPF, it's going to initiate mitosis. So if we look at it, here we have a graph of time versus relative concentration, and we also have up here something showing us when we're going through each phase of the cell cycle. Notice that when we start out here, we've got no cyclin. The amount grows until we reach our maximum peak, at which point you have enough MPF to make your cell move through mitosis. Then the cyclin is degraded and it's gradually built up again. Once it gets to a high enough level, you go through mitosis again, and then it's degraded. And if you have a cell that's constantly dividing, this cycle is just going to keep doing that over and over. How does it move your cell to the next part of the cell cycle? Well, here's some examples. MPF does the following. It phosphorylates and then by that activates proteins that are going to bind to the nuclear envelope and promote its breakdown. So they start breaking down the nuclear membrane so that way you can start sorting out your chromosomes. They also, some of the proteins that MPF activates promote the condensation of chromatin. So that way you've got these nice compact chromosomes you can pull around. And then just as a nice feedback thing, it promotes degradation of its own cyclin. So MPF, when it's present, inactivates itself. That makes sure that this signal gets turned off. So I talked about growth factors really quickly. These are an outside signal. So we have here our growth factor, which is acting, if you remember, from signal transduction like a ligand, binding to a receptor, which then sets off a signal transduction pathway, which can then go into, say, the nucleus and tell it, hey, make some cyclin, and basically initiate production of a cyclin. That would help move you to the next stage of the cell cycle. That would basically encourage the growth of cells. So there are lots of examples of these. You don't need to memorize any of them. You'll see, though, they'll be written out the first time you see them. And you'll also see GF at the end, which stands for growth factor. So you've got a bunch. There's TGF, beta, there's EGF, I think there's PDGF. I mean, there's a lot of growth factors. So now let's talk about when this goes wrong. When this, what happens during the cell cycle is you have a number of proteins that are in charge of making sure that not only did the cell do whatever processes it needs to prepare for division, but they're kind of a check. Well, you have these sets of genes. One of them is P53. A lot of them are Ps for some reason. And they negatively regulate the cell cycle. Basically, they turn off the cell cycle. So they're kind of like the opposite of the cyclin-CDK complexes. When they're functioning, if you have any of these conditions, something like DNA damage, some sort of abnormality in the cell cycle, hypoxia, which is really low oxygen, that's going to signal P53 to be activated or be made, and it's going to lead to cell cycle arrest. When that happens, you've got two different options. If you can fix it, you repair it and then keep going. If you can't fix the problem, then you undergo apoptosis, your programmed cell death. If there is something wrong with that, and P53 isn't working, and it doesn't respond to any of these signals, like, DNA damage or etc., then it's not going to stop the cell cycle. It's not working. So your cell cycle continues, and those cells that have reproduced can start gathering up and forming a tumor, and then you have cancer. So a lot of research on cancer focuses on cyclins, CDKs, and these tumor suppressor genes, because a lot of times the problem is you have a mutation in one of these tumor suppression genes, or in a cyclin or CDK that causes a cell to lose control of the cell cycle. Once a cell is through with the cell cycle, it can go into G0. This is a phase that can be lifelong for some cells, and for other cells, they can go in and out of G0 depending on the need. So there are some cells that are going to stop dividing completely. Most of your neurons and your muscle cells don't reproduce, which is why if you have like a heart attack and part of your heart muscle dies, you can't grow it back. Also, if you have damage to your brain, if you have a stroke, you don't grow those cells back. So those cells are in G0 and have been for most of your life. There are other cells that can re-enter the cell cycle, though, if they get the right signals and growth factors. So your liver cells, for example, if your liver is intact, probably don't need to do anything. But if your liver gets damaged, they can come out of G0, reproduce, and then repopulate your liver if they need to. So that is it. I hope it was enjoyable. Go study for your test.